Today we will see about the next cell organelle included under endomembranous system, which is known as Golgi bodies, and it is also known as Golgi complex or Golgi apparatus, and it is known as post office of the cell. Why it is called so, we will see here. And this Golgi body was first discovered by Camillo Golgi, and hence it is named so. From his name only, the Golgi body is named as Golgi complex or Golgi apparatus. So these are the densely stained reticular structure. So these are the densely stained reticular structure which is present near the nucleus. As in the last class you have seen, from the outer membrane of the nucleus, the endoplasmic reticulum arise. Okay, so the endoplasmic reticulum arise, which are of two types, RER and SER. So this RER and SER manufactures or synthesize the substances like lipids and fats. As you have seen, smooth endoplasmic reticulum synthesizes lipids, whereas rough endoplasmic reticulum synthesizes the proteins. So these synthesized substances are transported or given to the Golgi complex, and the Golgi complex modifies these substances, modifies the proteins and lipids. They pack up these synthesized substances and transport to different parts within a cell or between two cells. That means it helps either in intracellular transport or intercellular transport. So this is the main function. So means what? Main function means what? The substances which are being synthesized by endoplasmic reticulum are given or transported to the Golgi bodies, and Golgi body modifies these substances and helps in intracellular or intercellular transport, and hence because of this reason only it is known as post office of the cell. Okay. So this Golgi complex is also known as lipochondria or dictyosome. See, I have written on the right side as lipochondria and dictyosome. In case of plant cell, the Golgi complex is also known as dictyosome, and if it is rich in lipids, that means if it is the substances which are synthesized by SER are modified by Golgi complex, and if they are rich in lipids or fatty substances, we call it as lipochondria. So if it is asked the structure of lipochondria or dictyosome, it is same as Golgi body because it is the other name of Golgi bodies. Now the structure you can see here, the Golgi complex. So that means what? Golgi complex are also found in three forms. Okay, like as in endoplasmic reticulum, which exists in three forms like cisternae, tubules, and vesicles. Similar to endoplasmic reticulum, Golgi complex also exists in three forms, that is cisternae, vesicles, and tubules. That means the these long unbranched structures are cisternae. These are the vesicles, and tubules are also present, which tubules I have not drawn here. They are tube-like substances, and they are often branched. Okay, they are associated with cisternae only. And the next important thing is that the Golgi complex has two phases. Okay, so they have two phases. One is known as cis or forming phase, and this is the convex surface. Convex and concave you know well. So this is the convex surface, and they always face, it is always nearer to the nucleus. They always face towards the nucleus, and the other phase, the opposite to the cis or the forming phase, is the trans or the maturing phase, which is also known as the concave phase, and they face towards the plasma membrane. That means in a cell, this part face towards the plasma membrane, and this part face towards the nucleus. That means the cis or the forming phase or the convex surface, or and the other one is trans or maturing phase and the concave surface. That means the substances which are synthesized by endoplasmic reticulum are given to the cis phase or the forming phase. Hence they are named as forming phase. They modify these substances. See, they are released in the form of vesicles. They take up these substances and they modify it and released from the maturing or the trans phase. And one of the important cell organelle, which is the lysosome, that is also included under endomembranous system, are always formed from the Golgi complex. See, these vesicles which are released from the maturing phase is written as lysosome. That means some of the vesicles act as a lysosome also. So if you are asked for how from which cell organelle lysosomes are formed, that means lysosomes are formed from Golgi complex. Now Golgi complex is localized cell organelle. Okay, it is a localized, that means it is present in a particular area only, whereas endoplasmic reticulum is widely spread. So it is widely spread as it extends from the outer membrane and reach towards the plasma membrane, near to plasma membrane also. But Golgi complex is a localized one. That means it is present at a particular area only. And this Golgi complex has some functions, important functions. So I have written, you can see, number one is cell secretion. Cell secretion means that it is the chief function. Okay, it is the main fundamental function. That means Golgi body helps in secretion of some substances. That means it exports some macromolecules. Macromolecules means what? Lipids and protein. These are included under biomacromolecules. So they secrete and are exported. So secretion involves, that means 3 steps mainly. Number one is that it receives the material from the endoplasmic reticulum through the cis phase. Okay, so that means from this phase they will receive the material. They receive the material from the endoplasmic reticulum. Then they are modified. Okay, they receive the substances, they modify them, such as if it is protein, they are modified into glycoproteins. If it is lipids, they are modified into glycolipids. Okay, glycosylation or glycosidation of proteins and lipids is known as glycoprotein and glycolipid. That means glycosylation means along with the protein, some other substances are also present. So after chemical modification, they are packed into vesicles. So these are the vesicles. See, these are the vesicles. These are packed and given to the cis phase. They modify them again. They convert it into vesicles only. So after chemical modifications, they are packed into vesicles and they are pinched off. Okay, pinched off. We can also call it as exocytosis. See, these are pinched off. Means they tear off from the cisternae membrane and they are released outside and are sent to the part where they are required. So these vesicles that are pinched off are always from the trans phase or the maturing phase and discharged either within the cell or outside the cell. Okay. And because of this reason only, as I told, it is known as post office of the cell, or you can say middleman of the cell, or you can say director of macromolecular traffic in cell. Okay, middleman means what? They help in transporting of substance from one place to another, hence they are known as middleman. And second function, see, sorting, packing, export of material. That is, it is also same as what I told, that one only, that it receives the substances, modifies and export these materials to within that cell or to the other cell. Now third one is modifications of material originating of lysosome, peroxisome and glyoxysome. So modifications, chemical modifications of lipids and proteins, then originating of lysosomes, and other than lysosomes, these are the two cell organelles, peroxisomes and glyoxysomes. These are the other cell organelles which are also formed from this Golgi complex. Also, another important function is that it synthesizes the cell wall material. Okay, it synthesizes the cell wall material. Cell wall material means what? Number 5, synthesize cell wall material. So cell wall is present in plant cell. As I told in previous classes, that cell wall, which is seen in case of only plant cell, the materials which are present in the cell wall are synthesized by the Golgi complex, such as polysaccharide synthesis. So these makes up the cell wall and these are being manufactured or produced by Golgi complex only. And another is that formation of cell plate. Okay, so cell plate is what now? So say about the details study of cell plate we will see later on. But here just you remember, so in case suppose this is the plant cell, this is the cell wall, This is the cell membrane, this is the nucleus. Now when this this is the parental cells divide to form the two daughter cells in case of plant cell. Now when they divide this parental cell, there is a formation of cell plate. That means in the center region of this parental cell a plate is formed, okay, and this plate helps in division of the parental cell to two daughter cells. Later on this way they will divide, and this plate will divide the parental cell into two daughter cell, and these are known as these are the vesicles, okay. These vesicles, they arrange themselves at the center part. So these vesicles which they arrange themselves at the center part are known as phragmoplast, and these phragmoplasts are the vesicles which are produced by the Golgi complex. That means what? Vesicles of the Golgi complex forms the phragmoplast in the center of the parental cell, and they helps in dividing the parental cell in forming two daughter cell. That means this phragmoplast formed from the vesicles of Golgi complex helps in cell division. Hence, the other important function is that it helps in cell plate formation. So number 6 you can write as it helps in cell plate formation. And the last another important function is that it helps in formation of acrosome. Okay, helps in formation of acrosome. Now just you remember that acrosome is a part of sperm. Sperm means male gamete. So acrosome is a part of sperm or the male gamete which consists of some enzymes. Okay, enzymes. What type of enzyme if you were asked? Then hydrolytic enzyme. Hydrolytic means they helps in rupturing the outer wall of the female gamete. That means they helps in lysis. Lysis means what? Rupture or break or burst. So next we will start with lysosome. So see, it is the third cell organelle which is included under endomembranous system. So you have seen that lysosome is formed from, why it is called as included under endomembranous system, because it is formed from the Golgi apparatus. That means the vesicles which are released by the Golgi apparatus, some of the vesicles, they form the lysosome. And lysosome, it is also known as suicidal bag. It is also known as suicidal bags because we will see in the explanation why they are called suicidal bags. Okay, so these are the, if you are asked for the definition, membrane bound vesicular structure. Vesicular means the vesicles which are released from the Golgi apparatus. So it is a single membranous bound, like a nucleus is a double membranous. It is a single membranous bound vesicular structure formed by the process of packaging in Golgi apparatus. So it is present in all type of cells except in RBC. RBC is devoid of nucleus also, and it is also devoid of lysosomes and other cell organelles. So except in RBC, it is present in all type of cells. Okay, and in plant cell, the function of lysosome is carried out by the large central vacuole. The large central vacuole which is present in case of plant cell carries out the functions of the lysosome. That means lysosome is not present in case of plant cell. So now this lysosome, it is a spherical bag structure, single membranous bound to cell organelle, and the main important thing is that the lysosome consists of around 50 types of enzymes. What type of? Digestive enzymes. Around 50 different types of digestive enzymes, and they are known as, together they are known as acid hydrolase. Hydrolase means they will help in breakdown of some larger components into simpler ones. That is known as hydrolysis, and the hydrolysis process is carried out by these enzymes, hence they are termed as acid hydrolase, and they are acidic in nature, therefore the pH of these enzymes are around 5. So these enzymes actually are such as lipase, that will break down the lipids, proteins, that will break down the protease, and carbohydrates, for carbohydrates it is carbohydrase. Okay, so these enzymes, they are hydrolytic in nature. That means this will break down the larger into simpler ones. Like carbohydrate macromolecule will be transformed into the monomers, into its monomer. The next I have written, see, it is highly polymorphic. Polymorphic means what? They will change their shape. Okay, they can change their shape. They are polymorphic in nature. Okay, so these lysosomes, you can, we call it as suicidal bags. Now lysosomes are called as suicidal bags. Why they are called as suicidal bags? Because see, lysosomes consist of large numbers of enzymes, and due to the presence of these large numbers of enzymes, that means the hydrolytic enzymes, the large macromolecules changes into, or they are converted into simpler forms. And if this membrane happens to get broken, that means if they get burst or they get ruptured, the various cellular components, whatever means various cellular components means whatever the components or the structure or the biomolecule present within the cell would undergo lysis. Lysis means they will rupture, they will burst, and because of this nature they are known as suicidal bags. Okay, so first of all, it consists of large numbers of hydrolytic enzymes, that means digestive enzymes, and if they happen to get broken, the cellular the cellular constituents, that means the components of the cell, would undergo lysis. Hence they are known as suicidal bags. So this lysosome actually takes part in intracellular digestion of various types of material. That means the cell need to digest some materials, and these materials which are like synthesized by endoplasmic reticulum, packaging done by Golgi complex. Now they need to be converted into the simpler forms. So these simpler forms, the intracellular digestion, it is carried out by the lysosome. And the types of material may be endogenous, that means which are formed within the cell, or it may be exogenous, came from somewhere else means came from different cell also. So intracellular or intercellular digestion of exogenous or endogenous substances they carry out. Okay, so that is about lysosomes. Now we will see the types of lysosome. So see, I have written 4 types of lysosome. One is primary, secondary, residual, and autophagic. So primary lysosomes, they are also known as storage granule, and these primary lysosomes consist of only enzymes. So acid hydrolase enzymes, and they are always in inactive form. So next, if we see about the types of lysosome, so mainly there are 4 types of lysosome, which are known as primary, secondary, residual, and autophagic lysosome. So primary lysosomes are generally the newly formed lysosomes. So they are the newly formed lysosomes from the Golgi apparatus, which consist of only the enzymes, that means acid hydrolase enzyme. So they are newly formed from Golgi apparatus and consist of only the enzymes. Second is secondary lysosome, which are also known as digestive vacuoles or heterophagosomes. So digestive and vacuoles or heterophagosomes, because along with the primary lysosome, they consist of the digested material. So whatever substances like carbohydrate, proteins they digested, it consists of those substances. Hence, along with the primary lysosomes, phagosomes, they are known as phagosomes. So together we call this secondary lysosomes. Phagosomes means the digested part or the digested materials. That means the substances which are being digested by the lysosome. And third one is residual. Residual means it consists of the undigested material, the undigested materials which are later on released by the process of exocytosis. Exocytosis means the pinching of the materials. They get tear off from the membrane of the lysosome. So that is known as exocytosis, and these residual bodies or residual lysosome consist of only the undigested materials which are later on released by the process of exocytosis. Then the fourth one is autophagic lysosome. They are also known as autophagosomes. Okay, so autophagosomes, this contains the cell organelles to be digested. So cell organelles that need to be digested are carried out by these autophagic lysosomes with the help of the enzymes, and hence they are And since they digest its own material, own cell own structure, means own component present within that cell, hence they are known as autophagosomes. So these are about the types of lysosomes.